Hi everyone, my name is Caitlin Andrews, and I'm a PhD student at the University of Cambridge and Zoological Society of London in the UK. I've been very fortunate to spend the last 4 years studying this beautiful bird called the hihi, and today I'm going to be telling you a bit about their role as pollinators. But first, I just want to acknowledge some key collaborators that made this work happen. Sandra Anderson and Karen Vanderwalt for their pollination expertise, and to my supervisors, Rose Thorogood and John Ewan. Now, we can't talk about hihi without talking about translocations. 7 of the 8 hihi populations we have today are the direct result of these efforts, where birds are moved from existing populations to new sites in the hopes that they will flourish. Now, all you have to do is look at any of our reintroduced populations to see the success of translocations in the form of the world's cutest baby birds. But translocations have other goals. Hihi are important pollinators, so when we put them back into a habitat, we also hope that it will benefit native plants and the wider ecosystem. But while we know that translocations succeed in their species recovery goals, we don't know as much about how well they achieve these ecosystem recovery goals. So in this study, we decided to put our translocations to the test. Focusing on hihi's pollination of hihi, a common native plant, we conducted a pollination experiment across 4 sites. 2 sites where hihi had been reintroduced, and 2 sites without hihi. At each site, we selected 30 plants and exposed flowers to one of 2 conditions. Some flowers were covered with a wire mesh to keep birds out, so they were only pollinated by insects, and other flowers were left open, so they were pollinated by insects and birds. What did we expect to find? Well, we predicted that birds would improve pollination outcomes significantly at the hihi sites, while at the non-hihi sites, birds should improve pollination less significantly because our superstar pollinator, the hihi, was missing. We let the pollinators work their magic, and we came back later in the season to count how many fruits formed on each branch. And to our surprise, our results were the opposite of what we expected. Birds helped improve fruit set at the hihi sites, but they were even more important to pollination at the non-hihi sites. So we decided to take a closer look at what was happening inside the fruits. We collected a sample of fruits from each site, opened them up, and looked at their seeds. This gave us a sense of how many seeds were viable, meaning they formed an embryo. And we found that birds improved seed viability slightly more at hihi sites than non-hihi sites. There wasn't a huge difference, but this fit more with our predictions. Yet there was still one surprise here. Overall, seed viability was higher at the non-hihi sites, no matter if insects and birds were doing the pollinating. This suggests that there may be a particularly good insect pollinator at these sites, or perhaps something else is allowing the plants to produce more viable seeds, like better nutrients. But just because a seed is viable, it doesn't mean that it will actually germinate. So we let our seeds sit for 2 months in the lab and then counted how many viable seeds actually sprouted into seedlings. Here we found that birds improved germination rates significantly at the hihi sites, but hardly at all at the non-hihi sites. This suggests that hihi are improving the likelihood that seeds will germinate. To quickly recap, while hihi did not bring significant benefits to fruit set and brought only a small, if any, benefit to seed viability, they did help to ensure that seeds were more likely to germinate. Germination is a crucial stage of plant reproduction because it's ultimately what determines how many new plants are produced. So this shows that hihi bring unique benefits to pollination that other species may be less able to provide. That being said, we were surprised that there wasn't a greater difference in pollination outcomes between our hihi and non-hihi sites. Why might that be? Well, one explanation could be that ecosystems without hihi are simply less broken than we thought. Another pollinator could be filling in the gap left behind by hihi, or perhaps the plants themselves have adapted to making use of lower quality pollination. Meanwhile, ecosystems where we have reintroduced hihi may have other species present that are canceling out the benefits that hihi bring. For example, kakariki are known nectar robbers, which means they eat flowers without pollinating them, and they tend to be more common at hihi sites. But even if other species are keeping hihi from reaching their full potential as pollinators, hihi still have their secret superpower, which is their ability to improve seed germination. So when we translocate hihi, we need to remind ourselves that we're not just helping this one species. We're helping hihi help the ecosystem. Thank you to the following people for their support in the field. And finally, thanks to all of you for listening.